13.8 billion years ago, there was no matter, no space, no time, and no question anyone could ask about any of it. That is where this story starts. Not with life, not with Earth, not with chemistry. With nothing. Absolute nothing. No atoms, no light, no empty space waiting to be filled. The concept of before does not apply, because there was no time for anything to come before. And then, in a fraction of a second, so small, it has no useful comparison. Everything that would ever exist began expanding outward from a point smaller than anything you can visualize. That is not metaphor. That is the actual physical description of the beginning of the universe, and most people hear it and nod and move on without registering what it actually means. Here is what it means. The universe that emerged from that event was not complex. It was not rich with possibility in any immediately obvious way. It was hydrogen. Mostly hydrogen, some helium, trace amounts of lithium, and the laws of physics. That is the complete inventory. No carbon, no oxygen, no nitrogen, no phosphorus, no iron. None of the elements that make a living thing were present anywhere in the universe at this point. The building blocks of biology did not exist. The building blocks of the building blocks did not exist. Just the two simplest elements and an expanding void, cooling slowly over hundreds of millions of years. Now look at what you are. Your body contains roughly 7 octillion atoms. Most of them are oxygen, carbon, hydrogen, and nitrogen. Your DNA is written in a 4-letter chemical alphabet. Your neurons fire electrochemical signals across 86 billion cells. You are reading these words, and something behind your eyes is processing them, placing them in context, connecting them to things you already know. You are, by any reasonable measure, one of the most chemically complex structures in the known universe, and every single atom in your body came from that same expansion, that same initial inventory of hydrogen and helium in physics. The gap between those two states is the actual subject of this video, not a gap in the abstract sense, a specific, traceable, step-by-step sequence of physical and chemical events that took 13.8 billion years and produced, among other things, you. Each step in that sequence has been studied, most of it is well understood. And at several critical points, the honest scientific answer to exactly how it happened is some version of, "We are not entirely sure." That is worth stating clearly at the start. This is not a story with all its pieces in place. There are genuine open gaps, named, published, formally acknowledged gaps in the explanation. Those gaps are not where this story gets weak. They are where it gets interesting. Because the question of how a lifeless universe produced conscious beings is not a philosophical decoration. It is a real empirical question with a partial answer and several points where the partial answer runs out. We are going to trace this from the beginning. The Big Bang and the first elements. The stars that built the periodic table. The planet that formed from stellar debris. The chemistry that crossed the threshold into biology. The first cell. The 3 billion years of microbial work that made complex life possible. The nervous systems, the brains, and finally the part of the story that even the best science has not resolved. We are walking through the entire chain, and at each point the honest version is the only version worth giving you. The question is not whether the universe made you from nothing, because it did. The question is how, and the answer is stranger than most people have been told. In the first fraction of a second, the universe was not a place where anything we would recognize could exist. There was no space in any meaningful sense. No matter, no atoms, no molecules, no chemistry. What existed was energy at a temperature so extreme that the number requires scientific notation to write, and even that notation does not communicate what it actually means. We have no reference for it. Nothing in human experience maps onto a state of that density and heat. That is where this story starts. Then, in a fraction of a second so small it has no name in ordinary language, something happened that determined the shape of everything that followed. The universe inflated. Not expanded in the ordinary sense, inflated. A region smaller than a single proton stretched to roughly the size of a grapefruit in approximately 10 to the negative 32 seconds. If you need a number to anchor that, a proton is about a millionth of a billionth of a meter across. What this inflation did was distribute energy across space at a near uniform density, smoothing out irregularities, setting the conditions for a universe that could eventually form structure. Without it, gravity would have had nothing coherent to work with. The universe would have collapsed back on itself or dispersed into permanent formlessness. Inflation is why neither of those things happened. After inflation, the universe was still unimaginably hot, and it was cooling fast. At temperatures above 10 trillion degrees, matter as we understand it cannot hold together. Individual quarks moved freely. Quarks are the fundamental components of protons and neutrons, and in the first microseconds, they were not assembled into anything. They moved in what physicists call a quark-gluon plasma, a soup of particles interacting at energies that no particle accelerator on Earth can fully recreate. As the universe cooled below that threshold, quarks began binding together. 3 quarks per proton, 3 per neutron. The first stable pieces of atomic nuclei now existed, and the universe was roughly 1 second old. Here is what matters for the story of you. The window for nuclear fusion in the early universe was brief. As temperature dropped, there was a narrow range, roughly between 1 second and 3 minutes after the initial event, when conditions were hot enough for nuclear fusion to occur, but not so hot that it immediately broke apart everything it formed. Protons and neutrons could combine. Helium nuclei formed, some lithium. Then the temperature dropped below the fusion threshold, and that window closed permanently. What remained was fixed. Approximately 75% hydrogen, 25% helium, trace amounts of lithium. That ratio has been confirmed by measurements across the entire observable universe. It is one of the most precise predictions in all of physics, and the universe matches it exactly. Nothing else existed. That is worth sitting with. Carbon, the backbone of every organic molecule in your body, did not exist. Nitrogen, essential to DNA and protein, did not exist. Oxygen, phosphorus, iron, calcium, none of it. The universe had completed its first round of chemistry and produced 2 elements and a trace of a third. Every atom of every element heavier than lithium that exists anywhere in the observable universe had to be made later, by a different process entirely. The gap between that chemistry and a living cell is not a small gap. It is not even a large gap in the way we usually use that phrase. It is the gap between 2 hydrogen atoms bonded together and a human neuron firing. The universe had produced the former and had not yet produced anything else. What we have preserved from this early period is not nothing. Before nuclei could capture electrons and form stable neutral atoms, the universe was opaque, a dense fog of charged particles through which photons could not travel freely. This lasted roughly 380,000 years. Then the temperature dropped below about 3,000 degrees. Electrons bound to nuclei, atoms became neutral, and the universe became transparent. The photons that had been scattering endlessly through that fog were released, and have been traveling ever since. We detect them today as the cosmic microwave background radiation, a faint glow of microwave energy that comes from every direction in the sky, at a temperature of about 2.7 degrees above absolute zero. It is the chemical fingerprint of the early universe, preserved in light the same way a carbon isotope ratio is preserved in ancient rock. Every measurement of it confirms the same picture. This is not interpretation. The data is in the sky. After that moment of transparency, the universe entered a period cosmologists call the cosmic dark ages. There were no stars yet, but hydrogen and helium drifted in vast clouds, slowly responding to gravitational gradients seeded by quantum fluctuations from the inflationary period. There was no light source. There was no heat beyond the residual warmth of the background radiation, itself fading as the universe expanded. There was no chemistry happening that could build toward complexity. There was nothing that could metabolize, replicate, or respond. The universe sat in that state for somewhere between 100 and 400 million years, depending on when you date the ignition of the first stars. For hundreds of millions of years after that, the universe was dark and cooling, and almost entirely made of 2 elements, and there was nothing alive anywhere in it. The hydrogen sat in the dark for long enough that gravity found it. Not quickly. Hundreds of millions of years passed before anything changed. The universe was cold, structureless, and almost perfectly uniform. Almost. There were tiny variations in density, regions where matter was fractionally more concentrated than the surrounding space. Gravity amplified those variations. Dense regions pulled in more matter. They became denser still. The process compounded over millions of years until vast clouds of hydrogen were collapsing under their own weight, heating as they compressed, building pressure at their cores that the universe had never produced before. At a certain threshold, the temperature and pressure at the center of one of those collapsing clouds reached the point where hydrogen nuclei could no longer resist each other. They fused. The first star ignited. That is where the element problem gets solved. Everything after the Big Bang depended on this. The universe had hydrogen, helium, and almost nothing else. Those 2 elements are not enough to build a living cell, not even close. Carbon, oxygen, nitrogen, phosphorus, iron. These are the elements that biology requires, and none of them existed in meaningful quantities before the first generation of stars began burning. Stars are not just light and heat. That is the surface description, the poetic version. The accurate version is that stars are nuclear furnaces running a specific sequence of fusion reactions, and each step in that sequence produces a new element that did not exist before. Start with what happens inside a star. Hydrogen fuses into helium. That reaction powers the star and releases the energy we see as light. As long as there is hydrogen in the core to fuse, the star is stable. The outward pressure from fusion balances the inward pull of gravity. Eventually, the hydrogen runs low. The core contracts, heats further, and reaches the temperature at which helium nuclei can fuse. Helium fuses into carbon, not a metaphor for carbon. Actual carbon atoms, produced in the interior of a star. This is stellar nucleosynthesis, and it is the reason anything interesting exists in the universe. Carbon fuses into oxygen, oxygen into neon, neon into magnesium, magnesium into silicon. Each step requires more heat, more pressure, and only the most massive stars reach the later stages. The sequence continues up the periodic table, and in the cores of the largest stars it reaches iron. Iron is where fusion stops producing energy rather than consuming it. When an iron core forms at the center of a massive star, the nuclear engine shuts off. Gravity wins. The core collapses in less than a second. The resulting rebound is catastrophic. The outer layers of the star are expelled violently into surrounding space. A supernova. Here is what a supernova actually is, beyond the spectacle. It is a distribution event. A star spent millions or hundreds of millions of years building elements in its core, and then it explodes and seeds the surrounding region of space with all of them. Every element that star forged gets thrown outward at a significant fraction of the speed of light. The heavy elements synthesized in those final collapse moments, elements beyond iron that cannot form through ordinary fusion, are also produced and distributed. One supernova enriches the surrounding interstellar medium with the full periodic table of elements biology will eventually need. Now consider what that means for you. Your body is made of carbon, hydrogen, oxygen, nitrogen, phosphorus, calcium, iron, and dozens of trace elements. The hydrogen is original. Some of it dates to the first few minutes after the Big Bang. Everything else was built inside a star. The carbon in your DNA was fused from helium in a stellar core. The oxygen you are breathing was produced in the same sequence, one step further. The iron in your blood is specifically the product of stellar nucleosynthesis in massive stars. When Carl Sagan said, "We are made of star stuff," he was not being poetic. He was stating a fact in nuclear physics that most people never get told in its precise form. Your atoms have a prior address. They were inside a star. Then that star died and the explosion scattered them. What happened after that scattering is equally important. The material from supernova explosions does not disappear. It drifts through space as part of an enriched interstellar cloud, mixing with fresh hydrogen, cooling over time. Under the right conditions, gravity pulls that enriched cloud back together. The collapse of a molecular cloud, a giant mass of gas and dust seeded with heavy elements from previous stellar generations, produces a new star. Around that star, the remaining material forms a disk. Dust particles collide and stick together. Over millions of years, those accumulations grow into planetesimals, then into planets. That is how our solar system formed. Approximately 4.6 billion years ago, the Sun ignited at the center of a collapsing cloud of gas and stellar debris. The planets formed from what was left in the surrounding disk. Earth assembled from that material. Rock, metal, ice, organic compounds. All of it inherited from stars that had already lived and died before our Sun produced its first light. The water in your body, the calcium in your bones, the iron that carries oxygen through your blood. Each of these elements arrived here through that chain. Star formation, stellar nucleosynthesis, supernova, dispersal, new star formation, planet formation, and somewhere in that debris, the raw chemistry of biology. Every atom in your body heavier than hydrogen was forged inside a star that died before our Sun ignited. And the explosion that killed that star is the reason you have the raw materials to exist at all. The planet that formed 4.6 billion years ago from collapsing stellar debris was not hospitable. What you had was a molten rock, bombarded constantly by asteroids and comets, with surface temperatures that made liquid water impossible for extended stretches of time. No oxygen in the atmosphere. No ozone layer filtering the ultraviolet radiation pouring in from the sun. The air was methane, ammonia, carbon dioxide, hydrogen sulfide. Every gas that modern life would find lethal. That is the environment where biology was supposed to get started. This period has a name, Geologists call it the Hadean Aeon, named after the Greek underworld, and that name is not poetic license. It is a description. The moon was significantly closer to Earth than it is now, which meant tidal forces were far more extreme than anything we experience today. Volcanic eruptions were not occasional events. They were continuous features of the surface. There was no stable continental crust to speak of. What solid ground existed was repeatedly fractured, subducted, and remelted. If you are looking for a place that appears designed to prevent biology from taking hold, early Earth is a strong candidate. The bombardment phase is worth understanding specifically. Between roughly 4.1 and 3.8 billion years ago, a period called the Late Heavy Bombardment saw the inner solar system struck by an enormous volume of asteroid and comet material. The energy released by these impacts was sufficient to vaporize entire oceans, not damage them, vaporize them. Any liquid water that managed to accumulate on the surface would have been stripped away repeatedly. The surface temperature after major impacts would have exceeded the boiling point of water for thousands of years at a time. This is not a minor obstacle. It is a near-total reset of surface conditions, over and over again, for hundreds of millions of years. And yet here is what the geological record actually shows. Microscopic structures interpreted as fossilized microbes have been found in ancient rock formations in Western Australia, dating to approximately 3.48 billion years ago. Isotopic signatures that some researchers read as evidence of biological activity push that date back further, potentially to 3.7 billion years, possibly approaching 4 billion. The planet formed 4.6 billion years ago. The surface became broadly habitable, meaning liquid water could persist, somewhere around 4 billion years ago. That gives you a window of perhaps a few hundred million years between a barely stable surface and the first detectable trace of life. In geological time, that window is not long, it is remarkably short. What does that tell you? It tells you one of two things, and neither of them is comfortable. Either the transition from chemistry to biology is so probable that it happens almost immediately given the right conditions, or the evidence we have is misleading and life actually took longer than the current record suggests. Both interpretations are live positions in the field. We cannot determine with certainty which explanation is correct. Here is the difficulty with reading early Earth's geological record. Rock that is 3.5 to 4 billion years old is extraordinarily rare. Plate tectonics destroys rock. It cycles it back into the mantle, melts it, reforms it. The ancient formations that survive are small windows into a largely erased period. The Isua Greenstone Belt in Greenland, one of the oldest surviving rock formations on Earth, contains carbon isotope signatures that some researchers interpret as biological. Others argue the signal is the result of non-biological chemistry under heat and pressure. The rock has been through enough metamorphic processing that separating a biological signal from a geological one is genuinely difficult. Not impossible, difficult, and contested. What the record can tell us is the chemistry. Specifically, carbon isotope ratios. Living things preferentially use lighter carbon over heavier carbon in their metabolic processes, the same principle used to interpret the PETM fingerprint. When you find ancient rock with light carbon enrichment beyond what inorganic chemistry typically produces, that is a signal worth examining. Multiple ancient formations show this signature. None of them are perfectly preserved, all of them are debated, but the cumulative weight of that evidence is what pushes the estimated date of life's appearance back toward 4 billion years, and in some proposals, further. The surface of early Earth had one feature working in biology's favor, and it is the same feature that makes origin of life researchers interested in hydrothermal environments. Energy gradients, chemical gradients. Everywhere the Hadean surface was active, there were zones where hot met cold, where acidic met alkaline, where mineral-rich fluid met open water. Those interfaces are chemically productive in ways that calm, uniform environments are not. The same bombardment that repeatedly destroyed surface conditions also delivered organic molecules. Comets and carbonaceous asteroids carry amino acids, nucleobases, sugars. The same impacts that made early Earth a hostile surface were simultaneously seeding it with prebiotic chemistry from space. The raw material problem and the energy problem may have been partially solved by the same events that made the surface appear hostile to begin with. None of this closes the gap. Organic molecules delivered by comets are not life. Chemical gradients at hydrothermal vents are not life. They are ingredients and energy sources. What happened between those ingredients and the first self-replicating system is exactly where the chain of evidence breaks and the hard questions begin. The geological record says life appeared on Earth within a few 100 million years of the surface becoming habitable, which raises the question that every origin of life researcher is ultimately working toward. How? In 1953, a 23-year-old graduate student named Stanley Miller put methane, ammonia, hydrogen, and water vapor into a glass flask, ran electrical sparks through it, and walked away. A week later, the water in the flask had turned brown. Miller analyzed the residue. What he found were amino acids, 13 of them. The basic molecular units that proteins are built from had assembled themselves spontaneously from inorganic chemistry in a sealed glass container in 7 days. No biological process was involved. No enzymes, no cells, no pre-existing life of any kind, just simple gases, water, and an electrical discharge. That result changed the question. Before you can understand why that matters, you need to know what the field believed before Miller ran his experiment. The dominant assumption, largely unspoken but functionally embedded in how biologists thought about life, was that organic molecules were categorically different from inorganic ones. The word organic itself carried that implication. These were molecules associated with living things, produced by living things, requiring living things to exist. The idea that they could assemble from scratch in a non-biological environment was not widely modeled as a serious possibility. It was closer to a philosophical concession than a working hypothesis. Miller proved that assumption wrong in 7 days with a glass flask and a spark. The theoretical foundation he was working from was not new. In the 1920s, two scientists working independently on opposite sides of the world had arrived at nearly the same idea. Alexander Oparin, a Russian biochemist, proposed in 1924 that early Earth's atmosphere was chemically reducing, meaning it was low in free oxygen and rich in hydrogen-bearing gases. Under those conditions, he argued, ultraviolet light and electrical discharges could drive reactions between simple inorganic molecules, gradually assembling more complex organic structures. Across the Atlantic, British biologist J.B.S. Haldane reached similar conclusions by 1929, suggesting that early Earth's oceans, exposed to radiation and lightning, would have accumulated organic molecules over time. He called it, informally, a primordial soup, a body of water slowly thickening with chemical complexity over geological time. That framework sat mostly theoretical for nearly 3 decades. There was no obvious way to test it. Then Harold Urey, a Nobel laureate chemist at the University of Chicago, gave a lecture in 1952, laying out what the early Earth's atmosphere might actually have looked like, chemically. Miller was in the room. He approached Urey afterward and asked if he could attempt to run the experiment. Urey reportedly expected failure. He agreed anyway. What Miller built was elegant in its simplicity. One flask contained water, representing the early ocean. Another flask above it held the gases: methane, ammonia, hydrogen. Electrodes ran continuous sparks through the gas mixture to simulate lightning. A condenser cycled the water vapor back down, mimicking the water cycle. He ran it for a week and analyzed what collected. The amino acids were there: glycine, alanine, aspartic acid, glutamic acid, among others. The Oparin-Haldane hypothesis had, at minimum, passed its first serious test. The shock within the field was not immediate, but it was real. Some scientists were skeptical about whether Miller's simulated atmosphere accurately reflected early Earth conditions. That debate has continued, and it is legitimate. Later research suggested the early atmosphere may have contained more carbon dioxide and nitrogen than Miller used, which produces somewhat different results. When the experiment is rerun under those revised conditions, amino acids still form, though in different proportions. The core result holds. The atmosphere composition matters for the specifics, but the general finding has not been overturned. Organic molecules can emerge from inorganic chemistry. That is now established. What the Miller-Urey experiment did not do is equally important to understand. Amino acids are not life. They are not even close to life. They are molecular components, the way bricks are components of a building. Finding bricks scattered on the ground does not explain architecture. A living cell contains thousands of distinct molecular types, interacting in precisely coordinated ways. It has a membrane separating it from its environment. It has a mechanism for storing information and copying that information accurately enough for heredity to work. It has metabolic processes for extracting energy from its surroundings. None of that was in Miller's flask. What was in his flask was a starting point, a demonstration that the universe is capable, under the right conditions, of producing the raw materials. And that starting point matters more than it might appear. Before 1953, the distance between lifeless chemistry and the first living cell looked like an unbridgeable gap at both ends. The gap was still enormous after Miller, but its near end had moved. The building blocks could form on their own. The question shifted from whether organic chemistry could emerge without life to how those components assemble into systems capable of doing what living systems do. That is a harder question. It is also a more precise one, and precision is what makes a question answerable. The primordial soup model gave origin of life research its first serious experimental traction, and Miller gave it its first landmark result. What neither of them addressed was the step that comes after accumulation. A soup rich in amino acids, nucleotides, simple sugars. That is a chemistry problem. Getting those molecules to organize, link together, form chains, and eventually produce something that copies itself. That is a different problem entirely. The Oparin-Haldane framework described how the ingredients might appear. It did not describe how the ingredients start cooking themselves into something that reproduces. That question is where the next chapter in this story begins. Miller proved that the universe does not need biology to make the components of biology, which is a remarkable result, and it answered one question precisely while leaving the harder question completely open. The central problem with explaining how life began is that modern life is a system that requires itself to operate. DNA holds the instructions. Proteins carry them out. RNA moves information between the two. But DNA cannot copy itself without protein enzymes. Proteins cannot be built without instructions stored in DNA. And neither process works without RNA in the middle. Every piece depends on every other piece already being present. This is not a minor technical obstacle. It is a foundational logical problem. If you need all three components to run the system, and none of them can arise from scratch without the others already functioning, then how did the first one appear? This is the question that stopped origin of life research in its tracks for decades, and it is still the question that everything else in this field orbits around. Here is what the field eventually converged on. RNA can do both jobs. It can store genetic information the way DNA does, and it can catalyze chemical reactions the way proteins do. That dual capacity is not a minor footnote. It is the most important single fact in origin of life research. If one molecule can perform both functions, then the chicken and egg problem dissolves. You do not need DNA and proteins to already exist. You need RNA. Just RNA. A single molecular class that stores information and acts on it. In principle, that is enough to get something started. This idea has a name: the RNA world. The hypothesis proposes that before DNA, before proteins, before anything resembling modern biology, the first living systems ran on RNA alone. Self-replicating RNA molecules carrying genetic information and catalyzing their own reproduction. That is a striking claim. For most of the 20th century it was theoretical. Then in the early 1980s, the experimental evidence arrived. Thomas Cech at the University of Colorado and Sidney Altman at Yale independently discovered that RNA molecules could act as enzymes. Before that, the field assumed catalysis was the exclusive domain of proteins. Proteins were the workhorses of biochemistry. RNA was assumed to be a passive carrier. What Cech and Altman found was that RNA could fold into three-dimensional structures capable of accelerating chemical reactions without any protein involvement at all. These catalytic RNA molecules got a name: ribozymes. The discovery was significant enough that both researchers received the Nobel Prize in Chemistry in 1989. What does that tell you about what the field had previously assumed? It tells you the assumption was wrong, in a way nobody had seriously tested. Once ribozymes were established, the logic of the RNA world became much harder to dismiss. If RNA can catalyze reactions, and RNA can store information, then a self-replicating RNA molecule is not science fiction. It is a chemical possibility. Consider what that means: a molecule that carries instructions for its own reproduction and can carry out those instructions without external help. Once that exists, Darwinian evolution has something to work with. Variants that copy themselves more efficiently outcompete variants that do not. Small copying errors create variation. Selection acts on that variation. The entire machinery of evolution requires nothing more than a molecule that replicates itself imperfectly. RNA, in principle, can be that molecule. Now here is where honest science requires a more careful accounting. The RNA world is the most favored framework in origin of life research. It has substantial experimental support, and it still has gaps in it that have not been closed. The first problem is chemical fragility. RNA degrades. In the presence of water, it breaks apart. Early Earth had water everywhere, and any RNA molecules forming in an open aqueous environment would have faced constant degradation pressure. The conditions under which RNA could persist long enough to become biologically useful are not obvious. Some researchers argue that specific mineral surfaces or enclosing structures could have provided protection. That is possible. It has not been demonstrated in a fully satisfying way. The second problem is assembly. RNA is built from nucleotides. Each nucleotide is itself a complex molecule, a sugar joined to a phosphate group joined to a nitrogen-containing base. Getting those three components to assemble correctly under prebiotic conditions is not straightforward. Researchers like John Sutherland at Cambridge have shown plausible chemical pathways for nucleotide synthesis from simpler precursors. And that work is genuinely encouraging. But plausible is not the same as demonstrated. The specific environmental conditions that would concentrate and assemble nucleotides into functional RNA chains on early Earth remain a matter of active investigation. The third problem is perhaps the deepest one. Even granting that RNA nucleotides could form and link together, the jump from a short random RNA strand to a self-replicating RNA molecule is enormous. Self-replication requires that the molecule fold into a shape that can bind its own building blocks, position them correctly, and join them together in sequence. That requires a level of structural precision that is not going to appear in a random polymer. The question is, how many random RNA sequences need to exist before you statistically expect one with that property? Some estimates put the required number of random trials very high. Others suggest the problem is more tractable than it appears. Nobody has resolved this. What the field has done is build impressive experimental evidence for pieces of the RNA world. Ribozymes have been shown to catalyze reactions relevant to RNA replication. Short RNA strands have been shown to template the assembly of complementary strands. The building blocks can form under plausible prebiotic conditions. The pieces are there. What is missing is a continuous demonstration of the full sequence, from simple chemistry to a functional self-replicating system, operating without biological assistance. That experiment has not been done. Some researchers believe it is close. Others think the key step is still missing from our conceptual model entirely. Worth sitting with. That. 70 years of origin of life research, the best supported hypothesis the field has produced, and the central mechanism is still formally undemonstrated. That is not a reason to abandon the framework. It is a reason to be precise about what we know versus what we are inferring. The RNA World Hypothesis is the most favored framework in origin of life research, and it has the best experimental support, and it still cannot fully account for how you get from simple chemistry to a molecule copying itself reliably enough for evolution to begin. In 1977, a submersible exploring the ocean floor near the Galapagos found something that was not supposed to be there. The scientists aboard the Alvin expected cold, dark, barren seafloor. What they found instead was a community, tube worms several feet long, clams, crabs, shrimp, and bacteria, all thriving at depths where no sunlight reaches, under pressure that would crush unprotected human tissue, in water heated to temperatures that would kill most surface organisms outright. None of it was supposed to be possible. The assumption, held essentially without challenge up to that point, was that life on Earth runs on sunlight. Plants capture it. Animals eat plants. The chain begins with solar energy and nothing else. The Galapagos vents broke that assumption completely. Everything living at those vents was running on chemical energy, specifically on the energy released when chemicals in superheated water interact with cold seawater. No sun, no photosynthesis, just chemistry and heat and darkness. What that discovery actually meant took time to absorb. If life does not require sunlight, then the surface of a planet is not the only place life can originate. It also means that the origin of life on Earth may not have had anything to do with sunlight at all. Before 1977, the Miller-Urey framework dominated thinking. Lightning strikes an atmosphere, amino acids form, life eventually assembles in a warm surface pool or shallow sea. That framework assumes a surface origin. The vent discovery opened a second possibility, and over the following decades, a significant portion of origin of life research shifted toward it. There are two distinct types of hydrothermal vents, and the difference matters. Black smokers are the dramatic ones. Superheated water, sometimes above 400 degrees Celsius, blasting out of chimney structures built from mineral deposits. Highly acidic, loaded with metal sulfides, visually striking and chemically intense. But black smokers are now considered less relevant to the origin of life than the second type. Alkaline vents are quieter. The water they emit is warm rather than scalding, typically between 40 and 90 degrees Celsius, and chemically basic rather than acidic. They build pale, porous carbonate structures rather than dark mineral towers. The most studied example is the Lost City hydrothermal field, discovered in the mid-Atlantic in 2000. Lost City has been active for somewhere between 30,000 and 120,000 years. The structures it has built reach up to 60 meters tall, and the chemistry it produces is unlike anything found at black smokers. Here is the specific property of alkaline vents that researchers like Nick Lane and Mike Russell have spent years arguing is not coincidental. When alkaline fluid from the vent meets acidic seawater, a pH gradient forms across the thin mineral walls of the vent structure. Alkaline on one side, acidic on the other, a natural proton gradient, sustained continuously by the geology of the vent itself, requiring no biological machinery to maintain. That gradient is a source of energy, not metaphorically, chemically. Protons moving across a membrane from high concentration to low can drive reactions the same way water moving through a turbine drives a generator. Now look at your own cells. Inside every mitochondrion in your body, energy is produced by pumping protons across a membrane and then harvesting the energy released as they flow back. The mechanism is called the proton motive force. It is not a minor biochemical detail. It is the fundamental engine of cellular energy in almost all complex life on Earth. And the proton gradient that drives it is chemically identical in structure to the gradient that alkaline vents produce naturally, without any assistance from biology. Lane and Russell's argument is direct. The first living systems did not have to invent this gradient, they were born inside it. The vent provided the energy source. What life had to figure out eventually was how to carry that gradient with it when it left. The iron-sulfur chemistry of vent environments adds another layer to this. The mineral walls inside alkaline vents are rich in iron and sulfur compounds. These same iron-sulfur clusters appear at the catalytic cores of some of the oldest and most fundamental enzymes in living organisms. The acetyl-CoA pathway, one of the most ancient metabolic routes known, uses iron-sulfur clusters in ways that closely mirror the chemistry found inside vent minerals. This is not a loose resemblance. When biochemists map the catalytic structures of these ancient enzymes and compare them to the mineral chemistry of hydrothermal vent walls, the overlap is specific enough to suggest a direct chemical ancestry. The first metabolic reactions may not have required proteins to catalyze them. The mineral surfaces of the vent walls may have done the job first. Life may have started, not by solving the chemistry problem from scratch, but by inheriting a chemical toolkit that geology had already assembled. What this means for the origin of life is still actively debated. The vent hypothesis and the RNA world are not necessarily in conflict. They may describe different stages of the same process, or different aspects of the same environment. What the vent hypothesis contributes that surface models do not is a sustained, stable energy source and a natural compartmentalization structure. The porous mineral walls of alkaline vents create tiny chambers, small spaces where chemistry can concentrate rather than dilute, and natural geometry for prebiotic reactions that needed containment to proceed. The first protocells may have been vent pores before they were lipid membranes. The vent did not just provide energy, it may have provided the physical architecture that made chemistry into something more than chemistry. The gradient that powers the mitochondria in your cells right now is chemically identical to the gradient that alkaline hydrothermal vents produce naturally in the deep ocean, and that either means something important, or it is the most remarkable coincidence in biology. The gap between a self-replicating molecule floating in the ocean and a living cell is not a gap in degree, it is a gap in kind. A molecule that copies itself is a remarkable thing. But it is still just chemistry happening in open water, diffusing outward, diluting into nothing. The ocean does not care about your replication rate. Every copy you make drifts away. Every useful reaction you catalyze disperses into billions of liters of seawater and accomplishes nothing cumulative. For chemistry to become biology, something had to change, not gradually, fundamentally. A boundary had to form, inside had to become different from outside, and the moment that happened, everything that followed became possible. This is what the cell membrane actually is. Not just a container, a distinction. The membrane is what turns a collection of reacting molecules into a system with an interior, a chemistry that is concentrated and protected, and capable of accumulating complexity rather than losing it. Without that boundary, self-replication is just an interesting reaction happening in an indifferent ocean. With it, you have the first draft of a cell. You have heredity. You have the basic architecture that every living thing on this planet still uses. The question is, how that boundary formed in the first place. And the answer, like almost everything in this story, is stranger and simpler than most people expect. Fatty acids are among the most basic lipid molecules that exist. They have a water-attracting head and a water-repelling tail. When you put them in water, they do not dissolve evenly. They arrange themselves. The water-repelling tails cluster inward, away from the water, and the water-attracting heads face outward. The structure they form is a bilayer sphere, a vesicle, a tiny bubble with a membrane wall and a protected interior. No enzyme required, no genetic instruction, no biological machinery of any kind, pure chemistry, following the path of least energetic resistance, assembling a membrane spontaneously. This is not a metaphor or a simplified model. It is what fatty acids actually do in water under laboratory conditions, and it is what they would have done in the chemistry of early Earth. Jack Szostak's lab at Harvard has spent years pushing this further. His team has shown that fatty acid vesicles do not just form. They grow, incorporating new lipid molecules from their environment. They divide under gentle agitation, splitting into daughter vesicles. They take up RNA molecules from the surrounding solution, enclosing genetic material inside a protective membrane, without any active transport mechanism. None of this requires a cell. None of it requires evolution. It is the chemistry of lipids and water, doing what lipids and water do. What Szostak's work demonstrated is that the physical structure of a cell, the membrane, the enclosure, the capacity to grow and divide, can emerge from chemistry alone. The biology did not come first and then build the container. The container assembled itself, and the biology moved in. That enclosure changed everything downstream. Once RNA is inside a vesicle, its copies stay local. Useful reactions accumulate rather than dispersing. Vesicles that happen to contain more functional RNA grow faster and divide more. The ones that contain less do not. Natural selection does not require organisms. It requires differential replication. And once you have membrane-enclosed chemistry that copies its contents and divides, you have differential replication. You have the first mechanism that Darwinian evolution can act on. The threshold was not crossed by adding more complexity to open ocean chemistry. It was crossed by enclosing that chemistry inside a boundary that gave it somewhere to go. Now here is what that means for the history of life on Earth. Every living cell today, from bacteria to the neurons firing in your brain right now, carries the same basic architecture. A lipid membrane enclosing a hereditary system that uses the same genetic code, the same molecular machinery for translating that code into proteins, and the same chirality of amino acids. Left-handed amino acids, all of them, everywhere, in every organism that has ever been studied. This is not a design constraint from chemistry. Right-handed amino acids work perfectly well. The universal preference for left-handed versions is a historical accident, frozen into biology at a single point of origin. Everything alive carries that same frozen accident. What does that tell you? It tells you that all life on Earth, descends from a single ancestor, not a population, a single lineage. One cell whose chemistry became the template for everything that followed. Scientists call it LUCA, the last universal common ancestor. Not the first cell that ever existed necessarily, but the cell whose descendants survived and eventually became all life we know. We cannot find LUCA in the fossil record. It left no bones, no shell, no recoverable tissue. What it left is something more durable than any fossil. It left its molecular fingerprint in every genome that has ever been sequenced. We can reconstruct properties of LUCA by looking at what all living things share. It almost certainly lived in a high-temperature environment because the metabolic pathways most conserved across all life today work best at elevated temperatures. It had RNA-based heredity alongside early protein synthesis. It had a lipid membrane. It had the genetic code, or something very close, say to it, already in place. It was, by any reasonable definition, alive. Every living thing on this planet, every bacterium, every plant, every animal, every human who has ever existed, descends from a single cell that lived somewhere between 3.5 and 4 billion years ago, and we do not know precisely what it was or where it lived, but the molecular evidence that it existed is written into every genome on Earth. For the first 3 billion years of life on Earth, nothing you could see with the naked eye was alive. No forests, no oceans crawling with animals, no surface visible to anything that could observe it and recognize biology. Just rock, water, chemical gradients, and single-celled organisms doing chemistry in the dark. That is the story most accounts skip. They go from the first cell to the Cambrian explosion, as if the 3 billion years between them were a waiting room. They were not. They were the engine room. Everything that makes complexity possible, every nervous system, every brain, every thought you have ever had, required that invisible middle period to build the foundation first. Here is the number worth sitting with. The first cells appeared roughly 3.8 billion years ago. The first complex cell with a nucleus and internal compartments, what biologists call a eukaryotic cell, appears in the record approximately 1.8 billion years ago. That is a 2 billion year gap between the first living thing and the first cell that looks remotely like the cells in your body. 2 billion years of microbial chemistry producing, as far as the fossil record shows, nothing larger than a bacterium. The gap between a prokaryote and a eukaryote is not cosmetic. It is structural, and how that transition happened is one of the most important and least discussed stories in the history of life. The answer came from a biologist named Lynn Margulis, who proposed it in 1967 and spent years being dismissed for it. Her argument was straightforward and, to the mainstream at the time, almost offensive in its simplicity. She proposed that mitochondria, the organelles inside your cells that generate energy, were once free-living bacteria, not descended from bacteria, not similar to bacteria, actually bacteria, engulfed by a larger cell somewhere between 1.5 and 2 billion years ago, never digested, and eventually integrated so thoroughly that neither organism could survive without the other. The reaction from the scientific community was largely skeptical. That is worth noting. Her paper was rejected by about 15 journals before it was published. And she was right. The evidence is not ambiguous. Mitochondria have their own DNA, separate from the DNA in your cell's nucleus. They divide on their own schedule, independently of the cell dividing. Their ribosomes are bacterial in structure, not eukaryotic. Genetically, they are most closely related to a group called alpha proteobacteria. Every line of evidence points the same direction. What you carry inside every cell in your body is a bacterium. Ancient, integrated, indispensable. Not a metaphor, the actual thing. Two cells became one organism, and that merger changed the trajectory of life on this planet completely. Why does that merger matter so much? Energy. Before endosymbiosis, cells had a fundamental ceiling on how much energy they could generate relative to their genome size. Bacteria are efficient but constrained. When a cell absorbed a mitochondrion and kept it working, it gained an internal power plant that operates largely independently. The energy output per gene increased by orders of magnitude. More energy per gene means you can maintain a larger genome. A larger genome means more regulatory complexity, more sophisticated protein production, more behavioral range. Consciousness, as best we can tell, is an enormously expensive process in metabolic terms. The human brain uses 20% of the body's energy budget. That level of expenditure was simply not available to life before the endosymbiotic merger. The bacterium that got absorbed 1.8 billion years ago is part of the direct causal chain that ends with you being able to think about it. The other transformation happening in this invisible middle period is photosynthesis. Cyanobacteria developed the capacity to split water molecules using sunlight and release oxygen as a byproduct. From their perspective, if we are assigning perspectives to bacteria, oxygen was waste. It was the exhaust of their chemistry. From the perspective of the rest of the biosphere, it was poison. The atmosphere before approximately 2.4 billion years ago had almost no free oxygen. Life had evolved under those conditions, and the metabolic machinery of most organisms at the time had no use for oxygen, and was actively damaged by it. Then cyanobacteria started producing it in quantity. What followed is called the Great Oxidation Event, and it was, by any honest measure, a mass extinction. Organisms I could not adapt to rising oxygen. Concentrations died. The chemistry of the planet changed permanently. The atmosphere we think of as natural and life-sustaining was, at the time it formed, a catastrophe for existing life. But oxygen has a property that anaerobic metabolism does not. Aerobic respiration, burning fuel in the presence of oxygen, extracts far more energy from the same amount of food. The jump in efficiency is substantial. Organisms that evolved to exploit oxygen rather than avoid it gained an enormous metabolic advantage. And the mitochondria inside eukaryotic cells are precisely the machinery that makes aerobic respiration possible at the cellular level. The whole system connects. The cyanobacteria flood the atmosphere with a toxic gas. Most life is devastated. A subset of organisms evolved to use that gas as a fuel source. That metabolic upgrade, built on top of the endosymbiotic merger, is what makes the energy budgets of large, complex, behaviorally sophisticated animals achievable. No. What comes after? Not multicellular life, not nervous systems, not brains, is possible without these 3 billion years of microbial chemistry running first. The oxygen in the air you are breathing right now is the industrial waste product of cyanobacteria that lived 2.4 billion years ago. And the fact that you can breathe it instead of dying from it is because your mitochondria are descended from bacteria that evolved specifically to exploit what everyone else found toxic. 540 million years ago, the fossil record does something that troubled Darwin and has not stopped generating debate since. Before that point, the rock layers are largely bare of complex animal life. After that point, within a geological window of roughly 20 to 25 million years, most of the major animal body plans that exist today appear. Shells, eyes, limbs, bilateral symmetry, predators and prey, nervous systems. Darwin knew about this and found it genuinely disturbing. If evolution proceeds gradually, the sudden appearance of so many complex forms in so short a window looked like a gap in his theory. He said so directly. The gap has not fully closed. What the Cambrian explosion actually shows is not evolution failing. It is evolution accelerating. The question worth sitting with is why it accelerated then, after 3 billion years of microbial patience. The answer is not simple, and anyone who gives you a single clean cause is leaving things out. Several factors converged, and the convergence matters. Oxygen levels in the atmosphere had been climbing for over 1 billion years since the cyanobacteria began their work. By around 540 million years ago, those levels had reached a threshold. Active, muscular bodies with high metabolic demands require oxygen. Below that threshold, you cannot power the kind of sustained movement that predation and escape require. Above it, suddenly you can. The atmosphere had just become permissive for a new class, S of animal. At the same time, something was happening in the genome. Hox genes are regulatory sequences, master switches that control the development of body segments during embryonic growth. They do not code for specific structures. They determine where structures form and in what order. A small change in a Hox gene does not produce a small change in an organism. It can reorganize an entire body plan. By the Cambrian, the genetic toolkit for body plan variation was apparently in place, and the combinations available to evolution expanded dramatically. This is not a coincidence of timing. The molecular infrastructure for building complex, differentiated bodies had been assembled across hundreds of millions of years of microbial and early animal evolution. The Cambrian is when that infrastructure starts, producing results. Then add predation. This is where the ecological arms race begins, and once it starts, it does not stop. When one organism can eat another, the prey that survives is the one that detects the predator faster, moves away more effectively, or has a harder shell. Selection pressure sharpens immediately. Every generation filters for sensory capacity, for speed, for protective structure. The predator faces the same pressure in reverse. The prey that escapes is the one that was hardest to catch. So the predators that eat are the ones with better detection, faster movement, more precise control. Both sides are running the same race in opposite directions, and the pace of anatomical innovation accelerates under that pressure in a way that calm, resource-limited microbial competition does not produce. The eye is the clearest example of what that pressure produces. Eyes evolved independently at least 40 separate times across different animal lineages, not variations on a single inherited eye. Genuinely independent solutions to the same problem. Compound eyes, camera eyes, pinhole eyes, mirror eyes. Different optical architectures, different evolutionary origins, converging on the same basic function. What does that tell you? It tells you that the selective advantage of vision is so powerful that evolution found the solution over and over again, in completely separate lineages, across hundreds of millions of years. It also tells you something about the molecular toolkit. The gene PAX6, which controls eye development, is so ancient and so conserved that a mouse PAX6 gene introduced into a fruit fly will trigger eye formation in the fly, not a mouse eye, a fly eye. The instruction is so fundamental that even across that evolutionary distance, the signal is readable. The solution was always accessible. It just required the right pressure to be discovered. By the end of the Cambrian, the planet's biosphere looked recognizably animal in a way it never had before. Trilobites with articulated bodies and compound eyes. Anomalocaris, a predator more than half a meter long, with grasping appendages and large eyes mounted on stalks. Wiwaxia covered in scales and spines. Picaia, a small soft-bodied animal with a notochord, a stiffening rod along its back. that places it somewhere near the ancestry of every vertebrate alive today, which includes you. These animals were not simple. They had sensory organs, digestive systems, and the beginnings of centralized nervous systems. The basic architecture was present: sensory input, internal processing, motor output. That architecture is the foundation of every brain that has existed since. The universe at this point had been running for approximately 13.2 billion years. It started with hydrogen. It built stars, which built heavier elements. Those elements formed a planet. Chemistry on that planet organized itself into cells. Cells merged and differentiated over 3 billion years. And now, for the first time, there were bodies in the ocean that could sense their surroundings, process that information, and respond in coordinated ways. That is not a minor development. That is the universe building something genuinely new, not just complex chemistry, but systems that model the external world and act on that model. The Cambrian explosion gave the universe nervous systems, and a nervous system is a machine for processing information about the world, which means the universe had just built the first crude version of a tool it would eventually use to think about itself. The first nervous system was not a brain, it was a net. Jellyfish and their relatives had diffuse nerve networks spread across their bodies with no center, no hierarchy, no region in charge of anything. A stimulus hit one part of the net, and a signal moved outward. That was the entire system. It was enough to contract a muscle, to pull away from something, to coordinate a basic response. Not thought, not memory, just response. And that gap between simple response and what you are doing right now is the gap this section is about. The advantage of even a primitive nerve net was speed. Before neurons existed, cells communicated through chemical diffusion, molecules drifting from one cell to another. That process takes time. A neuron transmits a signal electrically, and it does so orders of magnitude faster than diffusion chemistry allows. The difference between those two speeds is the difference between an organism that reacts and an organism that does not. In a world where predation had just become a feature of the biosphere, that difference was everything. The Cambrian arms race did not just select for shells and claws. It selected hard for faster signaling. The next structural step was centralization. Flatworms were among the first animals to develop a recognizable concentration of neurons at one end of the body. Not a brain in any modern sense, but a ganglion, a cluster of nerve cells that processed more information than any other region. The body had a head end now. Information came in at the front, got processed in this small cluster, and signals went out to the rest of the body from there. That organizational principle, sensory input at one end feeding a central processing region, is the same organizational principle your brain runs on right now, scaled by hundreds of millions of years. The chordate lineage pushed this further. A spinal cord appeared, a protected channel running the length of the body, carrying signals in both directions between the brain and the peripheral nervous system. At the head end of that cord, tissue expanded and specialized. The earliest vertebrate brains were modest structures managing exactly what early vertebrates needed: body temperature regulation, respiration, balance, basic movement coordination. The brainstem and cerebellum handled these functions, and largely still do in you. When a physician checks your reflexes, they are testing circuitry that is roughly 500 million years old. It has not changed much because it has not needed to. Fish brains established the template. Amphibians and early reptiles built on it, without dramatic expansion. Then mammals appeared, and something new grew around the existing architecture. The limbic system, a set of structures sitting above the brainstem, became the seat of emotion and memory. Fear, attachment, the ability to remember that a particular place was dangerous, the motivation to seek out food and avoid harm. These were limbic functions. Mammals that could remember and feel had a survival advantage over those that simply reacted. Evolution retained the old structure and added around it. Nothing was discarded. The ancient brainstem is still in there, underneath everything, running the machinery that keeps you breathing while you think about other things. Primates added the prefrontal cortex. This is the structure that distinguishes the primate brain most sharply from other mammals. The prefrontal cortex handles planning, abstract reasoning, the capacity to model outcomes that have not happened yet, and the ability to represent other minds, to understand that other individuals have their own beliefs and intentions and knowledge states distinct from your own. That last ability, what researchers call theory of mind, is a cognitive tool with enormous social utility. A primate that could predict what other group members were thinking could form alliances, detect deception, and coordinate complex behavior. The selective advantage was not small. In the last 3 million years, the hominin brain roughly tripled in size. 3 million years is nothing in evolutionary time. The rate of change is genuinely striking. A brain that tripled in volume in 3 million years was under intense and sustained selective pressure. The metabolic bill for this is not trivial. The human brain accounts for roughly 2% of total body weight and consumes approximately 20% of the body's total energy budget. That is not an arrangement evolution maintains by accident. Structures that expensive persist only when the return on that metabolic investment is consistently high enough to justify it. Language, tool use, social modeling, long-term planning, the capacity to pass learned information across generations. These were the returns. Each one compounded the others. Greater language capacity enabled more sophisticated social coordination. More sophisticated social coordination created environments where more cognitive capacity paid off. The feedback loop ran for millions of years, and then, somewhere inside that process, something happened that the evolutionary account does not fully capture. The brain became more complex, more connected, more metabolically expensive. That much is documented in the fossil record. What the fossil record cannot show is the moment, if there was a single moment, when processing information stopped being just processing information. When there became something it was like to be the organism doing the processing. When the system stopped simply running and started, in some sense knowing that it was running. That question sits outside what evolutionary biology was built to answer. Somewhere in the evolutionary scaling of the primate brain, something started happening that goes beyond signal processing. And that transition, from a system that processes information to a system that experiences processing information, is the part of this story that biology can describe but has not explained. There is a point in this story where the standard scientific account hits a wall, and the honest thing to do is to say so directly. Everything covered before this point has a scientific framework, incomplete in places, contested in others, but a framework. Stellar nucleosynthesis is understood in detail. Abiogenesis has serious candidate mechanisms. Evolution is one of the most thoroughly supported theories in all of science. The emergence of nervous systems, the scaling of the primate brain, the architecture of human cognition, these are hard problems, but they are tractable ones. Researchers are making progress. The questions have address. This one does not. In 1995, philosopher David Chalmers published a paper that drew a line nobody in the field has successfully crossed since. He separated what he called the easy problems of consciousness from the hard problem. The easy problems are not actually easy, explaining how the brain integrates sensory information, how it directs attention, how it controls behavior. These are enormously difficult scientific questions, but they are in principle solvable by the standard methods. You build a model, you test it, you refine it. The hard problem is different in kind, not degree. Kind. Here is what it asks. We can identify every neuron that fires when you look at the color red. We can trace the signal from your retina through the visual cortex. We can map the network activity with increasing precision. None of that explains why it feels like anything. Why there is a subjective experience of redness rather than just a pattern of electrochemical signals processing in the dark. The technical term for this is qualia, the gap between the neural mechanism and the felt experience. And that gap, in 30 years of serious scientific and philosophical attention, has not closed. That is worth sitting with. The standard physicalist position is that consciousness will eventually be fully explained by neuroscience. Once we understand the brain in sufficient detail, the subjective experience will follow. The assumption is that consciousness is what the brain does, the same way circulation is what the heart does. Map the mechanism completely enough, and the mystery dissolves. This is not an unreasonable position. It is the working assumption of most neuroscientists. But it has a problem. Every advance in mapping neural correlates of consciousness gives us more detail about the mechanism. It does not give us any purchase on why the mechanism is accompanied by experience at all. The gap is not shrinking. It is just better described. Integrated information theory, developed by neuroscientist Giulio Tononi, attempts a different approach. It proposes that consciousness is a measurable property of any information processing system, defined by how much integrated information that system contains. The measure is called phi. High phi means more consciousness. In principle, this makes consciousness quantifiable and potentially universal. Any system with sufficient integrated information would have some degree of experience. The theory has genuine mathematical rigor and serious proponents. It also has critics who argue it produces conclusions that seem physically wrong, and that the framework, however elegant, does not actually solve the hard problem, so much as relocate it. Panpsychism gets dismissed in popular discussion as mysticism. It is not. It is a serious philosophical position held by serious thinkers at serious institutions. The core claim is that experience, in some form, is a fundamental feature of reality, not an emergent product of complex biology, but a basic property of the physical world, the way mass and charge are basic properties. The argument is not that rocks are conscious. The argument is that the standard materialist framework has no account of how purely non-experiential physical processes produce experience at any level of complexity, and that if we cannot explain the transition, perhaps there is no transition. Perhaps experience goes all the way down, combining and integrating into the rich conscious experience we have at the level L of a brain. Philosophers like Philip Goff have argued this case with rigor. It is not settled. Nothing in this territory is settled. What we do actually know is this. Anesthesia research has revealed which brain systems appear necessary for conscious experience. The thalamocortical system, the dense interconnection between the thalamus and the cortex, seems critical. When anesthetics suppress activity in this network, consciousness disappears. When activity returns, so does experience. Disorders of consciousness, from vegetative states to minimally conscious states, have given researchers clinical windows into what happens when normal brain integration breaks down. The neural correlates are being mapped in increasing resolution. We know more about the structure of the problem than we did 30 years ago. My position is that knowing the structure of a problem more precisely is not the same as solving it. The hard problem is not yielding. What is genuinely unresolved is whether our current conceptual framework is even adequate to address the question. Some researchers argue that the hard problem is a product of the way we frame experience, that with better concepts the mystery will dissolve. Others argue the opposite, that consciousness is precisely the kind of phenomenon that resists third-person scientific description, because it is, by definition, a first-person phenomenon. Science is built on intersubjective verification. Consciousness is irreducibly subjective. Whether those two things can be reconciled, and how, is not a peripheral question. It is the central question that the chain from hydrogen to here ultimately runs into. You are 13.8 billion years of physics and chemistry asking itself what it is, and the universe has not found a way to answer that question yet. And the fact that the question can be asked at all is either the most remarkable accident in the history of everything or the point the whole process was building toward.